Hello, my name is Cecilia Sumajo, and first of all, thank you all of you for tuning in today. The seed constitutes a turning point in the evolution of land plants, and it is a salient feature of all seed plants. It is morphologically very diverse, as you can see here, due to ontogenetic changes in the seed coat, but also to associated structures like the caruncle in a rhizome, the trichomes of Strelitzia, and the ariel of Taxus. Gnetales, a small order with around 110 species, has a stunning yet intriguing morphology, and the seed is covered by associated structures that we're going to refer to as envelopes. We work with three different species, Gnetum and Ephedra antisyphilitica, both with fleshy seeds, and Ephedra californica with completely dry seeds, allowing us to have a better understanding of the development of morphologically diverse seeds. So the seed is a fertilized ovule, which is formed by a nucellus or megasporangium covered by the integument here in yellow. So most gymnosperms have only one integument covering the nucellus and orthotropous orientation, whereas most angiosperms have two integuments, inner and outer integument, and anatropous orientation. It is important here to highlight that it is still unclear how the ovule evolved. So there are three main hypotheses to explain it. One suggests that the ovule evolved as a new structure. Another one suggests that it is by fusion of telomes. And a third hypothesis suggests that the ovule evolved by sterilization and fusion of sporangia. So, as I mentioned before, not all species have the typical ovule morphology described for the major groups, and it turns out that Gnetales is one of the exceptions. The ovules have the integument, here in yellow as well, and one or more extra structures that have been often interpreted as outer integuments. But they are not completely similar to the integuments of angiosperms, starting by the fact that in angiosperms, the inner integument develops first and then the outer integument. But in Gnetales, the outer one develops first, and also by several morphological and structural differences. So, following recent literature, we're going to call it envelope. Because of its features, Gnetales enriched the debate on the evolution of seed plants. Based on morphological analysis, Gnetales has always been considered a sister group to angiosperms. But phylogenetic analysis never retrieved this relationship. And in fact, Gnetales is always found within conifers. In terms of ovule development, what we know the most is from angiosperms, specifically the model species Arabidopsis thaliana, for which we know the genes that are involved in integument initiation, ovule initiation, as well as integument planar identity, so that flat identity of the integuments, and the genes involved in integument elongation as well. So, having a better understanding of the morphology, anatomy, and development of members of this group will contribute to our understanding of the evolution of the seed, which is our focus with this project, but also of the evolution of seed plant lineages. So, we were able to collect cones of Gnetum throughout their development in order to describe their morphology and their anatomy. Some of these cones are bisexual cones with the ovules growing on top of the microsporangia or the pollen cones from the same node. But some cones are also unisexual. So Gnetum is well known to have three envelopes or three structures covering the nucellus: the integument, middle envelope, and outer envelope. But this is only for the unisexual cones. On a bisexual cone, the ovules have only two structures covering the nucellus: the integument and one extra envelope. As the seed matures, the outer envelope becomes fleshy and red. The middle envelope becomes sclerenchymatic, forming the stony layer, and the integument compresses, forming a papery layer. So we look also at the expression patterns of the candidate genes throughout ovule development in Gnetum. In this cartoon here on the left, we can see the expression patterns of the gene Integumenta in the ovules of Arabidopsis at two different developmental stages. Integumenta is a gene known to be involved in integument development in Arabidopsis. In Gnetum, we detected expression of the Integumenta homologue in the nucellus throughout the entire nucellus, as well as in the apical region of the integument. BEL1 is also a gene that is known to be involved in integument initiation in Arabidopsis and integument development. And in Gnetum, we saw expression in the nucellus as well as in the megaspore mother cell. Canary genes are very well known for their function in the abaxial-adaxial identity of the integuments in Arabidopsis. But interestingly, in Gnetum, one of the copies is expressed throughout the nucellus and the megaspore mother cell, and the other homologue is restricted to the megaspore mother cell and the apical region of the integument. Unicorn is also a gene that is involved in the flat identity of the integuments in Arabidopsis. And in Gnetum, there are two homologues as well. One is expressed throughout the nucellus, and the other one is restricted to the megaspore mother cell, and again to the apical region of the integument. So being able to perform this analysis in Gnetum allowed us to identify that all these genes have different expression patterns compared to what we know from Arabidopsis. But in fact, none of them are expressed in the envelopes of Gnetum. So this allowed us to provide molecular evidence suggesting that the envelopes from Gnetum are genetically different to the integuments in angiosperms. So the question now is which genes are involved in the development of these structures in Gnetales. So to answer this question and to have a better insight into the genes that are involved in the development of these structures, we sequence the transcriptomes of the different tissues in Ephedra: the young cones, the shoots, pollen cones, ovules, bracts, which are these envelopes, everything separate. And we did it in these two species. Both have the bracts or envelopes covering the integuments. And in Ephedra californica, these envelopes remain completely dry as the seed matures, whereas in Ephedra antisyphilitica, they become fleshy as the seed mature. The structural differences between these two species are evident since very early in the development of the ovule. In Ephedra californica, we see that the parenchyma cells forming the bracts are completely compressed and sometimes collapsed, giving them this papery appearance since very early in the development of the ovule. Contrary to the cells that are found in the bracts of Ephedra antisyphilitica, which are more isodiametric. The cuticle is also thinner and with less ergastic substances. So these differences appear since very early in the development of the ovules in these two species. We also detected major differences with the transcriptome analysis. So with for Ephedra californica, we identified 407 differentially expressed genes, from which 23 are transcription factors. And we detected many MIP-related genes and histone 4s that are known to be involved in ovule development arrest. And in Ephedra antisyphilitica, we detected 524 differentially expressed genes, from which which 34 are transcription factors. And interestingly, many of the upregulated genes are members of the ethylene-responsive transcription factor family, which are genes known to be involved in fruit ripening in fleshy fruits, as well as Matchbox genes like TT16 and agamus, which are known to be involved in ovule development and have been often referred to as being involved also in this fleshy seed characteristic. All right, so from this work, we can make four major conclusions. One is that there are differences in the integument developmental network across seed plants. And one of the striking differences that we detected is with the polarity genes. So these genes are known to be involved in the adaxial-abaxial identity. But in Gnetum, we're seeing something completely different. We see these genes restricted to the apical region of the integument. So this suggests that they may have a function more in the proximal distal plane of the ovule. We also notice that all of these integument genes are expressed in the sporangia, similar to what is known for mosses and lycophytes. And so this suggests that these genes were co-opted from a sporangia developmental network, but also provides molecular evidence for the origin of the ovule as a result of sterilization and fusion of sporangia. As mentioned before, we still don't know the genetic mechanisms involved in the development of the envelopes, but it is not by by the integument developmental network. And these led us to identify novel genes involved in their development in other species, like in Ephedra californica and Ephedra antisyphilitica, and the genes identified here definitely deserve further characterization. Finally, I think all of us here are aware of the importance of working with different and non-model species. These studies allow us to have a better understanding of the evolution of plants in terms of morphology, anatomy, and development, and in general to have a better understanding of the evolution of plant lineages. I would like to thank my home institutions, the New York Botanical Garden and the City University of New York, and all our funders. Also to the Botanical Society of America for all the support I received during my graduate studies, and to all the botany organizers for their amazing job with these meetings. And thank you all of you for being here, and I'm happy to take questions anytime. Thank you.